Hello, everyone. We are back live today. It is Thursday, 12:30 p.m. Mountain Standard Time, and we are here with All Things AuD. I'm Dr. Natalie Phillips.
I'm Dr. Hannah Galloway.
And I'm Trista Hauser-Smith.
All right. So, what was fun, what we thought would be fun to just kind of cover are different research spotlights. And this was the brainchild of Dr. Hannah Galloway because she loves to look at different research articles. And so she had sent us this earlier this week. And I thought it was a really cool research article. And so, on All Things AuD, we're going to continue to have these research spotlights. So I just wanted to let people know if there were interesting articles that you came across that you want us to read and research, take a look at, maybe discuss, then please feel free to send it to us because I think it will make Dr. Galloway very happy. So anyway, so this particular research that we're going to talk about today is an article that was done. And I'm going to share it in the links in just a second here. But this was a research that was done by Dr. Matt Stewart out of Johns Hopkins. And they were really looking to see if COVID is directly damaging cells of both the middle and the inner ear. And so I thought it was really interesting, actually. And what they found is that they first had to look at how are they going to do this, right? And so they looked at these surgical tools that they could use because they were really concerned that if they use particular surgical tools or drills, right, that there would be these particles of this virus that are going to be all over in the air. And so it would be very hard for the researchers to kind of stay safe. And so we thought that this was kind of an interesting article. And I'm going to let Dr. Galloway kind of talk about the tools that they used.
Yes. So Dr. Stewart, who actually studied geochemistry in graduate school, which I thought was hilarious because I'm like, oh, you went to graduate school for geochemistry and then also became an MD and then also got a PhD after that. So anyway, he just kind of thought that he could use this mineral saw because it wouldn't blow particles around like, typical bone saws would. And at Johns Hopkins, historically, they've collected samples, from a really long time ago of cross sections of the inner ear. And so they kind of already knew that this was a possibility and thought of it as a way to study this. So what they did is they carved a hole in the temporal bone that's as wide as a spaghetti noodle, so 2 millimeters. And they used hand tools. One was called an osteotome, and the other were curettes, which they described as really sharp, tiny ice cream scoops. So they scooped a little tiny spaghetti-sized hole in the temporal bone and then put a swab up there to swab and see if they could find COVID bacteria or COVID virus in the middle ear. And they actually were able to find that. So they found the genetic signature of COVID in middle ears of 2 of the 3 cadavers. At first, they were only given 3 COVID positive cadavers to look into. And then it said in an unpublished follow-up research, COVID was found in 60% of the middle ears of 20 cadavers. So they did, in fact, find it directly in the middle ear. It sounds like they weren't terribly shocked because we know COVID infects the nose and, the mucus and everything. It's not all that far apart. So I thought that was pretty cool. Just how they had to do it to, be conscious of the fact that they didn't know how COVID could spread and protect the researchers. So that was kind of what they found out about the middle ear. And then they wanted to push it further and look at the inner ear. So I'll let Trista tell you guys about that.
So what we know basically in research is that sometimes it's very hard to look at a human skeletal system or inner ear, things like that, because ethic issues, we can't just chop people up as they're living. And so there's a lot of things that they had to tackle through this. And so through research, we've done a lot of research in many fields with mice because they have a lot of things that are very similar to humans. And so we can compare and contrast with some of those. And so there was a study in 2020 that did actually find COVID receptor cells, or what they call ACE2, in the inner cells inner ear cells of mice. And so Dr. Stewart actually looked at that research and was like, well, maybe we can try to replicate this somehow in humans. Because I wanted to kind of determine if that COVID existed in the inner ear, but that's really hard to do because the inner ear is very tiny, very delicate parts, very hard to see. And so cadavers are a great way to analyze that. So they did use what they call a diamond saw, very, very tiny things so they could chop up little pieces in these cadavers of the inner ears and try to figure out if that COVID virus was actually in some of these ears. And like Dr. Galloway was talking about before, they had lots of sections from the 1800s that they had looked at. You can see in this picture right here, just other models looking at the inner ear. So they were trying to replicate that in a way. So their preliminary results looked at and found that the receptor sites that COVID could actually attack areas in the middle ear and the cochlea. And that this could also affect the balance system in the cadavers who did not have COVID. So they were just seeing that the receptor cells that for the people that did not have COVID were present, but then those that did have COVID, could see how they were attacking those areas as well. So really cool research just by being able to look at cadavers that were actually donated to this place and that they could cut up really tiny. And you can see in this picture here how tiny of those little things that they got from the inner ear. Really cool. So
yeah, it's incredible to think about slicing something that thin. And because I nerded out about this article, I looked up the kind of saw that they used, and it said in the article that they couldn't get the NIH to fund the purchase of the saw because it was experimental and it cost $7,000. So they had to get like private funding for the saw. But I looked it up, and it looks it didn't look like what I expected. It looks like more of like a band saw or like something that you used in shop class. But the blade is like really, really thin and little, and has lots of little teeth on it. So I'm guessing that's, what made it possible. But
Yeah, I love this picture. And this is out of the article that we put out in the comments as well. So it's a clickable link that you guys can get to this article as well. But I love this picture because you can actually see the semicircular canals on that right side, right?
yeah,
And so that's how thin they did this slice. It is incredible. But when I saw that picture, I was like, holy cow. So not only did they, use that diamond mineralogic saw, right, to make those little slices, but like to be able to put it between the glass to put under a microscope is what you're looking at, right? I just think it's really, really cool. And so I'm going to put up one other picture as well, because it really takes a look at the slides and how they actually had to store them. And I thought that was kind of really cool too. So let me see if I can get this one up as well.
I'm also imagining, so you're a researcher and you know that no one's done this sort of technique since the 1800s. And Johns Hopkins only gives you 3 cadavers. And the inner ears are so tiny. Wouldn't you be scared that you would mess something up?
Oh my goodness, yes.
absolutely.
Okay.
Yeah, stakes are high.
Okay, so this is kind of how they stored it. And so there you have it, those slides that are still accessible, which is crazy also to just think about that, is that how accessible that is. And so this article, these were pictures from the actual article, and we thought that was really kind of cool to see how they use these cross sections, these very, very thin cross sections to study that. And I was just fascinated too by even thinking about not using a particular drill because it could get those particles in the air and to really protect those researchers. So I thought that was kind of interesting, and that they had to go back to the 1800s, I believe, right, to figure out, well, we used to do it like this, and let's try it again. And
to think that they have like a whole library of samples. I think it said they had over 5,000. I can't remember, but anyway, that historically they just have like that room is just full of cross-section samples, which is pretty crazy. I'll also mention that the article that we are going to post, it's from undark.org, which is not a typical site that I would post like peer-reviewed research from, but I thought they did a really nice job of explaining, how they did it and why it's different and why it's important. And there are links to the actual, peer-reviewed journal articles too, if you have access to that.
Yeah, awesome. Well, great article, Dr. Galloway. Thanks for finding it. And again, if you are coming across something that's interesting or you wonder if there has been research on it, let us know. Send us a direct message because we can take a look at it and review it and see if it's something that we want to discuss. And we'll thank you for it as well, too, because we definitely want to continue to have these research spotlights. We think it's really interesting. So with that, we are going to sign off. We're going to remind people to be back here next Thursday. We have a very, very special guest on with us next Thursday at 12:30 p.m. Mountain Standard Time. You do not want to miss this guy. And we'll look for it in some of our hints on some of social media to see if you can guess who it's going to be. But it's going to be some guy who has just some amazing audiologist who is on his way to retirement. So we're going to have that discussion with him next week on February 3rd. So we'll see you then. Thanks, guys, for joining. All right, we'll see you.